everyone. I'm very happy to have been invited to give this talk on pollinator-mediated gene flow between crops and their wild relatives, the case of the brinjal. We have domesticated thousands of crops globally, and these crops have been derived from wild plants. So over 1,000 plant species are closely related to the world's most important food crops. So this is very important for our food security. So according to the literature that I've scrutinized, India has 166 species of native cultivated plants and 320 wild relatives. So crop wild relatives, which the acronym is CWR, contain adaptive genetic variation missing from domesticated crops. And they're very important for developing high-yielding varieties, pest resistance, and for enhancing nutrient content. So the message is that the loss of genetic diversity of wild genome will have adverse consequences for plant breeding. So today I'm going to talk about the brinjal, Solanum melongena, which is an ancient vegetable crop, very familiar to Indians because it's native to Asia and has been domesticated in India, China, and Malaysia. Actually, this is still not clear, but it's definite that it's been domesticated in India. It's been cultivated since prehistory, and the earliest written record is from Sanskrit and from China. There are a diversity of cultivars in India, but the trouble is this diversity is reducing because land races are being substituted by F1 hybrids. So the diversity of our vegetable crops are also going down, and the danger of this is there be homogenization due to genetic erosion. So this is a picture I got from the web on the diversity of brinjals. I'm sure there are more than this, but it gives you an idea. So the brinjal was the progenitor of our crop brinjal was Solanum insanum, which is the wild brinjal. So Solanum insanum is distributed in Madagascar, India, Southeast Asia, and China. It grows widely in the plains of India around villages. I'm sure many of you have seen this plant. It's a thorny perennial, which has got small greenish fruits, which turn yellow eventually. It forms an interbreeding complex with cultivated brinjal. So this has been known for quite some time that this is the progenitor of the cultivated brinjal. So this is the distribution of Solanum insanum, Madagascar, Indian subcontinent, Southeast Asia, and China. So what are the similarities and differences between crop and wild brinjal? The crop is an annual plant, while the wild is a perennial plant. In fact, it lasts for 10 years. I've seen some plants here which are still surviving. The crop is mostly non-spiny, whereas the wild is a spiny, low-growing bush. The crop brinjal comes in various fruit sizes and colors, whereas the wild brinjal has small greenish white or white fruits, and the ripe fruits are yellow or orange. In fact, the tribal people used to eat this wild brinjal very frequently in the past. Now, I think it's no longer popular with them. The flowers are violet or purple, and very similar between both. And pollen is the only reward. And the brinjal crop, as well as the wild, are andromonaceous. That means René described the breeding systems of plants. Andromonaceous means one plant has got male flowers and bisexual flowers. That's hermaphroditic flowers on the same plants. And both are buzz pollinated by solitary bees. So one issue is that when you introduce any crops with trans genes, you have to do a risk assessment, especially if wild relatives occur together. Because what happens is due to hybridization and backcrossing, crops genes can go into the wild relatives. And if it's a transgenic crop, then this transgene gives the wild relative a huge fitness advantage, which turns it into a competitor of the crop in turns to a yield. So gene progression causes wild to be more like the crop. And enhanced weediness in the wild relative growing along crops is due to the higher fitness conferred by the transgene. So a very classical example is the weedy red rice that has been described, I think, in many parts of the world, particularly China. So this rice competes with the crop. It looks exactly like the crop and competes with the crop and contaminates the harvested seed because the rice is red in color. So you can't even sell the seed. It's not a uniform type of rice seed. So transgenic herbicide-resistant weedy rice can destroy production due to higher fitness. So in many places, they've given up growing this transgenic rice because the competition from the weedy relative. Another outcome is that there could be an extinction of the wild relative. This has been also documented. Not many studies have been done on this, but what happens is the genotype is swamped by the pollen from the transgenic crop and eventually goes extinct. There's also an impact on non-target organisms. For example, butterflies that feed on pollen, bees, all these pollen-feeding organisms can be affected by this Bt gene in the pollen. And this has also been shown in the case of monarch butterflies in the United States. I mean, it's not been clearly demonstrated, but there are indications that it can affect their survival. So there are some questions before introducing transgenic crops like the Bt brinjal. So first of all, you have to examine whether the genetically modified crop has extant relatives in the site where it's going to be introduced. What is the extent of natural hybridization between the crop and the wild? What is the current ecological role of the wild relative? So here we conducted a study with some colleagues in the United States and France to see whether there's potential for gene flow between the crop and the wild brinjal. So these are some of the questions that we asked. That is, do wild and cultivated brinjal occur near each other? Do flowering periods overlap? Are wild and crop brinjal self-compatible? Does wild brinjal rely on pollinators with seed set? Do wild and crop share pollinators? Can pollen from cultivated brinjal set seed in the wild relative? And documentation of gene flow between crop and wild. So we selected 23 sites for the field survey to see whether crop and wild occur together. So this is from the Karnataka and Tamil Nadu. And it's along the eastern flank of these mountains because the wild brinjal generally occurs at lower altitudes in dry areas. And the green square is no crop within 0.5 kilometers, and the red triangle is crop within 0.5 kilometers. And so we did a survey to see whether crop and wild brinjal occur together. And of course, in many places, they certainly do. So out of 23 sites, 61% had crop and wild within 0.5 kilometers. We selected 0.5 kilometers because this is the average flight range of a solitary bee. And do they overlap in flowering times? Of these 14 sites, in 9, that is 64% of the time they overlapped in flowering times. That means there's potential for pollen flow between the crop and the wild. So one aspect we looked at was the style length, because when the style is long, it prevents selfing, because the pollinator here in the wild brinjal on the left here, it has got a long style. So the pollinator touches the style first, deposits pollen, and then goes on to sonicate the anthers through buzzing. And here's the crop. So the wild and the crop look similar, but the style is very short. Both are buzz pollinated by solitary bees. So here you have this amygdala, which is a solitary bee, buzzing a crop flower. And you can see the cloud of pollen emerging from the anthers. This is a xylocopa bee, also a solitary, the carpenter bee, which also is buzzing this flower. And this is nomia, a halictid bee, which also buzzes flowers. So they certainly do share pollinators. For example, these three pollinating insects, Nomia, Amygdala, and Xylocopa. They're all buzzing bees, and they buzz flowers, and they both visit wild as well as crop. And the pollen collection in these flowers were high, and they contacted the stigma. Whereas some other bees also visited these flowers, and there was a sylphid fly as well. And Heterotrigona is quite successful in crop brinjal, but Apis is not. The Apis bees were no good in pollinating the brinjal. So we looked at the outcrossing rates in wild and crop brinjal. So when you have flowers that are naturally pollinated, open pollinated, here you have 72% fruit set in the wild and 41 in the crop. This 41 is maybe because it was close to a suburban area with fewer pollinators. And then self-pollination. What we did here is we enclosed the flowers to see if they could set seed by self-pollination. And the wild doesn't set any seed by self-pollination, and the crop very low levels of seed set. And then we hand self the flowers and then enclose them in the bag. And here again, wild low, and crop also, fairly low. And hand crossed, that means we got pollen from a different plant and manually pollinated these flowers. And in the wild, it was a fairly good fruit set. And then we did a crop into wild hybridization, and 23% fruit set was obtained in this hybridization experiment. Work. And here you have a hybrid fruit, you see, spiny, it's got a sort of greenish in color. And here you have an F1 plant. We had to destroy all this after the experiment. But you can see that they can hybridize, that they can set fruit. And we also did a genetic study to assess the gene flow. And here we took 10 sites. One in sort of fairly central and northern Karnataka, the first batch of sites, and then you have the Nilgiri Biosphere Reserve here. And then you have the southern Western Ghats, that's Kalakkad and sites in the south. So we did the genetic diversity assessment with 14 SSR loci. This was done by my colleague, Dr. Alison Snow in the United States. So we found that the wild populations are genetically variable. That means in different regions, you have different levels of genetic variation. And all this was statistically significant between sites, all the measures of genetic variation. Whereas the crop populations were more uniform. Here you have the measure total alleles, mean number of alleles, private alleles, expected observed heterozygosity, inbreeding, and this is the outcrossing rates. So both crop and wild are capable of outcrossing. And then we looked at the genetic structure and differentiation of the wild populations, and measured the FST, which is the fixation index, which measures levels of population differentiation. So the average the global FST was 0.48. That means they were highly differentiated, and it ranged from very low level to very high levels of differentiation. So among population percentage of the analysis of AMOVA, analysis of molecular variance, which measures the levels of differentiation, is 46%, and within populations it's 54%. Then we looked at isolation by distance. So there was increasing population differentiation with geographic distance, which indicates that we have a lot of diversity in the wild populations, which have to be conserved because it's not homogeneous like the crop. And then this is the genetic structure of the wild and the crop plants. Here you have mainly three structures. And here, these are the populations that we measured. It goes from the first one in Karnataka, it moves from Karnataka, southern Western Ghats. And we had three crops among them from Karnataka. So here you can see that the first wild population is very, very closely allied with the southern Western Ghats population. And this is probably because these wild brinjal seeds are considered to be medicinal. So they're harvested and traded. So probably some seeds from here have gone to Karnataka and have been cultivated there or germinated or whatever. And then here you have the second kind of genotype, which is a mixture of crop and wild. So here you see in this region, which is in Karnataka towards the north, that there's a lot of genetic mixing between the crop and the wild. And here you have the Nilgiri biosphere populations. They have some substructuring, but they're fairly uniform. And then the southern Western Ghats populations, the blue one towards the right. So the first cluster consists of populations from the Nilgiri Biosphere Reserve. This is quite a sort of distinctive cluster. And the second cluster is from the southern Western Ghats. And one population from Karnataka is found here, allied with the southern Western Ghats. And the third cluster is from Karnataka towards the north. And here we had three different crop plants growing next to these wild plants. And here you find that there's a lot of gene exchange between the crop and the wild. So what we concluded is that wild and cultivated brinjal tend to occur together. They overlap in flowering times. They tend to be outcrossing. They share pollinators. They can form F1 hybrids. And there's ongoing gene flow between the crop and the wild. So if you want to look at the publications, there are 2 publications that came out of this. And then we also looked at the pest attack, because the rationale for introducing transgenic brinjal into India is that it reduces attacks by pests. And the major pest is this moth larvae, Leucinodus ordinalis. So we looked at the level of infestation of wild and crop brinjals, 164 plants and 316 plants here. And 54% of the crop brinjal were infested by these lepidopteran pests, all 3 species, majority by this Leucinodus ordinalis, but none of the wild brinjal. In fact, we even did an experiment where we had some larvae and gave them wild brinjal leaves, and none of them developed. So obviously the wild brinjal plant is resistant to attack by these pests. So the implications for conservation, I mean, we now know that pollination and seed dispersal is very important for the food security of global food security. But we also have to understand that preserving the wild relatives of these crop plants is very, very important, because they have genes that are no longer present in the crop domesticated plants. So if we need any improvement or anything, we need to preserve these heritage populations. So what we conclude is that crop to wild gene flow is evident, and there's a high degree of divergence of wild brinjal populations in southern India. And this indicates that we have a high diversity. If you look all over India, probably we have a high diversity of wild brinjal germplasm resources, which need to be preserved either in situ or ex situ. So here we can see the population 1 and 2 are suitable for in situ and ex situ germplasm conservation, whereas population 3 has multi-local genotypes, most similar to cultivars. So if you introduce BT brinjalia, it is quite likely that the trans gene is likely to escape into wild populations. And we don't know what it's going to do there, because it's an experiment that is waiting to happen. And so the only way to conserve these resources is to isolate target wild populations through proximity to crops to preserve the wild germplasm. So we need an urgent measure to go and identify different wild brinjal populations in the subcontinent, and then try to see which can be protected from pollution from crops, especially if these are transgenic crops. I mean, with wild crops, obviously, it's not as serious as when there's transgenic crops which are applicable. So thank you very much, Renee, and the organizers for giving me this opportunity.
A question from Uma Shankar, who asks, are the wild into crop seeds fertile?
Wild into crops, yeah. That means when the wild pollen is deposited, it goes two ways. But the thing is, when the wild is the donor, the crop fruit doesn't develop much. So they are interfertile, but the seeds, likelihood of getting fertile seeds is not likely. Whereas with the other way around, when the crop is the donor, pollen donor, then you get fertile seeds at one point.
Uma Shankar has another question for you. What is the overlap in bee visitation between the wild and crop plants in nature?
We were looking at crops side by side with wild, but it is in the same region. This is within 2 kilometers, crop and wild. It's not adjacent to each other, but within 1.5 to 2 kilometers. So here you see that the overlap is quite high. And
right. So would you say that the apists, for example, were also visiting the wild, but and maybe stealing pollen? So once the anthers have been sonicated by, say, something else, is there a likelihood of pollen to be stolen from?
There is. Yeah, they do visit the male flowers, and try to collect pollen. But
after it has been sonicated by a legitimate. Okay.
But they prefer, for some reason, they prefer crop plants. Crop brinjal, yeah. Wild brinjal is very rare, but common.
Uma Shankar has another question. He says, when you say wild, do you mean land races?
No, it's not a land race. It's a wild,
Right. So I think you had mentioned that if you go back to your slide where you showed a picture of the wild species. I think you.
This is the wild species. It's a very short kind of straggling plant, and this is the distribution.
This one, right? So okay. So Rajesh Tandon has a question to you.
Yeah.
And he says, what is the extent of pest attack on F1 or natural hybrids?
We didn't look at that because our research ended with the F1 hybrids. We didn't look at the pest attack on them. In any case, but to preserve them because of ethical concerns, we just destroyed them.
Absolutely. But the fact that you found a lot of the pests on the crop, and actually your table said 0 pest attack on the wild, right? The same pests were not.
Same pests.
You were looking at the same pests, not
same pests, same region actually, side by side.
Okay.
The crop and the wild were from the same kind of area, and the crop.
I see.
Yeah, and they're still growing around. I can't see a single pest on them, but they are eaten by spotted deer and cows, the wild plant.
Okay, so that they are also probably contributing to the seed dispersal aspect.
Seed dispersal, they contribute to seed dispersal.
Right, which is absolutely unstudied in.
Unstudied, yeah. I think we need to do more work on these wild relatives of crops.
Absolutely, and
preserve them.
I think your point is absolutely well taken, and I'm so happy that you've brought this to the fore, particularly with our sensitivity to GM crops and the brinjal having been at the center of all the controversy. But I think that your data really gives us additional food for thought.
I just wanted
Somebody
one
else?
because here is a situation.
Shivana, please go ahead. Shivana.
Because your wild species is so resistant to pests, whereas cultivated is so susceptible. Is somebody working on hybridizing this and transferring genes or
Nobody does
anything you are
any
aware
research
of?
on this. In fact, they remove all the wild plants,
No, my point is, any agriculture universities or horticulture universities, are they doing any work? Because you said more than
I don't think
50%
anyone
of
is
the
doing
plants get infected with the pest.
Right.
And whereas the wild species
Unfortunately, yeah.
But this is a straightforward case. If somebody can try to transfer this gene, identify this gene, and so on. So you are not aware whether there is any work going on in this direction.
I don't know of any work, but it could be used for crop improvement of the crop.
Yes, certainly. That's what I.
It's a very important resource. Yeah, and when I read more about the transgenic crops, I get a bit more anxious because
right. I absolutely agree with you, but I think Shivana has also brought up this point that our agricultural universities and our whole concept of, investigating these important problems is just not there. So we need to really thank you so much, Priya, for joining us despite, all the issues that you are having.
I'm really sorry that I am missing.
No problem. It was a very, very insightful talk. Thank you.